This presentation on SuperMASSA is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 13, 2021.
Welcome to the SuperMASSA training section. And my name is Marcelo Molinari. I'm from, actually I'm from Brazil, but I work in North Carolina State University. I'm a senior research assistant there. And today we're going to talk about genotype calling and ploidy estimation using SuperMASSA. The first thing I would like to ask you is to download this folder if you want to follow along with the analysis. But and everything we need is within this folder, which is in our shared drive. Just let me. Where is the chat? Here. Here. So I will share this link here, and if you guys want to just download this folder and unzip it, okay? So yeah, so today I will present a brief introduction to the model. The SuperMASSA model is just an intuition because we don't have the time to go deeper into the statistics of it. And in the first part, we will use this model and SuperMASSA to estimate ploid levels in sugarcane by parental population. And in a second part, we'll perform a genotype calling in a sweet potato by parental population using another software called VCF2SM. Just some words before I start. So SuperMASSA was developed with a specific question in mind. How can we infer ploid levels in such a complex species as sugarcane? So the first question that we wanted to answer when we implemented SuperMASSA was to estimate ploid levels. And of course, if you estimate the ploid levels, you can do the second thing you ask is what's the genotype of each one of the individuals in the population for each SNP, right? So the basic idea of SuperMASSA is to rely on this, as Roland showed us, is rely on these angles here, which are the expected places of each one of the dosages, depending on the ploid level. And for this particular case, we are assuming that we don't have any bias towards the reference or the alternative allele, right? So when in our original data set from sugarcane, we used mass spectrometry. It's a very particular technique to access SNPs, but it's very precise, and I think this works great with that technology. But I have some experience with also sequence data, which works well as well. So in this first figure here, we have the expected angles for an hexaploid. In the second figure, we have the expected angles for the octaploid, and so on, decaploid and dodecaploid, right? So, of course, if I remove these colors and I plot these lines behind these points, of course, a trained eye can answer this question, right? But we need a probabilistic way to do this. And the way we choose to do this is using a Bayesian network. A Bayesian network is just a connection of random variables, and they are connected through these arrows with a conditional probability. So basically, when you have two nodes and you have an edge or an error, connecting, you have a dependence between, in this case, for instance, we have ploidy and we have the genotypes, and here the genotypes depends on the ploidy level, and D are the data depend on the genotype, and so on. And when you have this network, what you try to do is to, given the data, given your observations, which we call evidences in these probabilistic graphical models, we try to maximize this model. And maximize is actually choose the best configuration for each one of these nodes. And the probability that you need to maximize is the joint probability of this network. And what we can do to obtain this probability, we can use a bunch of conditional probabilities based on this network. So this is a model-based approach. And for each one of these conditional probabilities, we have a model. And then we use all these probabilities together to get this joint probability. And what we are going to try to do is to maximize, and actually what SuperMASSA does is to maximize the posterior configuration of this network, right? So, and this is just a naive explanation. If I show you guys these graphics here, and I say to you, oh, I have my individuals in this plot, and what is the best configuration given that I have a biparental population and my ploid level is 8. In the graphic that seems to be more appropriate to explain that phenomenon or that configuration, that scenario, is this one. And what SuperMASSA does is try to, it finds a probabilistic way to find the best configuration between all possible configurations. And when it finds the best configuration, means that the SuperMASSA found the best ploid level that explains the configuration, the genotype of the parents, and also the genotype, the dosage call for each one of the individuals in this population. And with that, similarly to what Roland presented, we also can have the posterior probabilities for each one of these individuals, right? So let's say we have this individual here, and originally it was classified as red or as 1 dose or 7 dose, because here we have an octaploid, right? But how can we compute the probability of this individual belonging to other classes? What I can do is to set this individual or assume that this individual is in a different class and recompute the probability of the model. And then the second way, I can set this individual to a second class and compute the probability, and to a third class and compute the probability. And I can normalize these probabilities, and we will have a similar probability distributions as the ones that Roland showed us. But this, we call this naive probabilities, because actually we are fixing a model, we are assuming that we have a model, and we just vary one of the individuals in the model to obtain these probabilities, right? But we can obtain this using SuperMASSA as well, okay? This was a very, very brief introduction, because otherwise we will not have time to go through the scripts. And yesterday we had a lot of problems when installing Python 2.7. And what I did here was to resuscitate a Windows machine I had here, and I installed Python. And this was literally, these were the 3 steps that I took to install. I downloaded Python from this location, and then I changed my directory to Python.7 scripts and installed matplotlib, and it was ready to go. For Mac, you can try to do this. I used in my Mac, and for Linux, you can try to do this. But here, I'm assuming that you have Python 2.7 installed, up and running, okay? If you have any questions, we can discuss in the Slack channel. But for now, let's assume that we have Python 2.7, okay? Okay, so let's go to the first demonstration using a single instance of SuperMASSA to estimate ploid level in sugarcane. Just let me enter in my RStudio. There you go. I have here my RStudio. And I'm going to use RStudio, but I'm going to use the terminal feature in RStudio. As you can see here, this is my Linux machine, and I can use this terminal instead of using the terminal in the machine. If you can't see this terminal, you just go in Tools, Terminal, New Terminal. You have a terminal here, okay? And on that folder that I asked you guys to download, you unzip it and you enter in this folder, and then we have these three folders. I'm going to choose ploid estimation, and then I'm going to open ploid in sugarcane.sh. The first thing we need to do is to change our directory to the ploid estimation folder, which is, for me, is in this location. I will change. And then, just to check what we have here, we have here our scripts. And if you are in Windows, you use dir instead of ls. And basically what we do is to use Python, invoke SuperMASSA, and then we have several arguments. And I'm going to explain each one of these arguments here. So Python is our program, the language that SuperMASSA is written. Here we have the script, supermassa.py. You need to know where this script is. You need to point to the correct location of this nib of the script. And here we have the genotypes. We have two types of arguments here. So usually these commands work like this. You have the language, in this case Python 2.7, the script, and several arguments. This is very common in Unix world. And we have two types of arguments. The arguments with no values. So in this case, just I'm saying to the program, just print the genotypes. And we also have arguments with values. In this case, we have the type of the inference we are going to use. In this case, I have a biparental population. I'm going to use F1. We have Heidi-Weinberg as well. And if you want to use with no structure, if you will, we use just the argument ploid. So instead of F1, you use HW or ploid, depending on what type of inference you want. In this case, F1. And here, we need to choose a ploid range. Here in sugarcane, the literature says that we have from 6 up to 14, 16. And in this case, just to be more fast, I will choose from 6 to 12, okay? And when I use this type of syntax here, 6 column 12, I'm saying to SuperMASSA, please analyze 6, 8, 10, and 12, just the even numbers. If you want the odd numbers, you can do something like this. You can put a step here. So in this case, it will go from 6 to 12, with steps with increments by 1. So you have 6, 7, 8 up to 12. But in this case, just let us use the even ploidy levels. And here is a threshold for the individuals, for reporting the individuals with a certain posterior probability. So in this case, we are choosing 0.8 to only keep individuals with associated probability of 0.8 or higher in our output. Okay. So if you want all our individuals independently of the posterior probability, you just put 0 here. Okay. Here we have our file, our input file, and our input file. Just let me show you the input file. In this case, as I said to you, this is a mass spectrometry file. But in a second part, we are going to show some VCF files. And basically, you have the 2 intensities here and the name of the individuals. And for the parental file, we have the number, the name of the parent, and here is also the intensities for each one of the allelic variants. And that's it. And let's run this command here, and let's see the output. Yeah, there you go. So in this case, we have some probabilities here. So this is the log likelihood for the whole model, the joint model. And this is comparing all possible ploidy levels that we used. So we used from 6 to 12. And this is the probability of this particular SNP is 8. Okay. And in this case, we have these parameters here are the dosages of both parents. In this case, we have 0, a nulliplex, and a simplex. Or if you are choosing the other allele, we have 8 doses and 7 doses. And here we have the genotypes, or the genotype calling for each one of the individuals in the population. If you are running in Windows, you can try to run this command here. It's a little bit different, but it will produce the same result. Another way to run this is instead of just running a huge line here, you can split the line split the command in several lines using the backslash. Okay, and in this case, I'm saving in an HTML file. Just let's run this as well, and it's easier to see in the HTML file. Okay, so here we have the same results, right? But just in a prettier way. Okay, yeah. So this is, we run just for one SNP, right? And it's usually we want to run for several SNPs. And what I have here is a script in R that actually uses R to run SuperMASSA using this very special command called system. Okay, and now I'm going to use R. As you will see, I will change this to console here instead of terminal. I will load these 2 libraries here, ggplot2 for graphics and dplyr for handling the data set. And I'll just set my work directory to ploid estimation. And then what I can do is to write the exact same command I wrote here in this system command within R, and it will show the same result. Actually, this is another file, right? I'm using another SNP here, but the idea is the same. And using this intern equals true, I'm running something outside R and I'm bringing to R. And if you want to run this in Windows, we have these commands here. And as you can see, this is pretty clunky. It's not a beautiful output. And what I did here, I just program a small function that just to try to make it more, just to capture the important things in this result. And if you are going to use this in your data set, you just make small adaptations. If you want, we can discuss this, but this is just a function to parse this output. And then I will run this function here. And in R, when you want to do a certain job several times, you usually implement these functions, right? And here we have 3 arguments in this function. I created this function with the result from this SuperMASSA analysis, and both files from the offspring and the parental file. And if you run this function using the result, you have a graphic like this. So this, and this is the graphic, similar to what Roland showed us. And in this case, we have a decaploid, right? And the probability associated to that ploid level is 0.98. It's a very good probability, but very high. The dosage of the first parent is 3, and the second parent is 0. So this parent here, the red one, is 0. Yeah, the triangles are the parents. I forgot to say that. And here we have the population. Each one of the dots is one individual in the parental population. Since we can do this for one file, the great thing of being within R is that we can do the same thing over and over using a loop. Of course, you can do this in Bash or in other languages in your terminal, but I think this is easier because we are more used to use R, and that's what I did here. So the first thing I do is to list all possible files that I have. And just let me show you, oh, this bar keeps in my way. Okay, so in this case here, we have the sugarcane data files. We have 27 different SNPs here, just as an example. And I use this function list files to list all these files. And as you can see here, I have all files in this directory, or in this folder. And then I will separate the parental files and the offspring files and the progeny files using this function called grep. And I will use this function to select the names that with a pattern par and the pattern pro. And if you see here, I just have the parents, and here I have the progeny. Okay, so 2 different sets, because we need the combinations of the same SNP and the same data from the same SNPs from parents and progeny. And what I'm going to do here is to actually use this for loop. And for those of you that doesn't work with R and loops, this is a very simple structure and very, very useful and powerful, because you can use a particular variable here, let's say I, and I say, well, do this thing for all values in between 1 and the length of this vector. So do this thing here with I, 1, 2, 3, and so on, okay? So if I just put I equals 1 here, I will have this. I'm just pasting this script. I'm just pasting the name of the files, and I'm building this source code here. Oops, I'm sorry, src, which is basically the same command we use in the terminal, okay? But I can do the loop to access different files. And then I run this in SuperMASSA, and after that I can print the SNPs and obtain the temporary file here, this with the ploid level, the probability associated to that ploid level, and the dosages for each one of the parents. And what I'm going to do here is to run this loop, but instead of printing on the screen, I opened this PDF device, and instead of just showing on the screen, it's saving in this PDF file. So we have 27, and I tested this in Windows as well, but instead of using this command here, you would need to change to this command here, right? Because, and actually what you want to do is to check if your Python is in this particular path here. Usually when you install Python 2.7 in Windows, this is the usual path, but you need to make sure to point to the right direction. And here we go, we have our 27 markers. And just let's check this, the results. And here we have, yeah, there you go. We have the results for each one of these SNPs. And here we have the ploid level and the associated probability to this ploid level. This is a very interesting one. You see, we have a very low probability because, as you can see, there's some biases here, and the cloud is not exactly in the expected angle, so SuperMASSA can detect that. But other ones are better. You see 6, and this is from the same biparental population. Okay. I don't know, but sugarcane usually has in the same individual, you can have multiple ploid levels. So that's one of the greatest complications in sugarcane, and very high ploid levels. And just to summarize here, and I forgot to say that I use this df variable to concatenate all the results that I got in this loop. So here in the first column, we have the probability, the ploid associated, and the dosage of both parents. And what you can do here is to plot a box plot with all possible ploid levels for this particular population. Okay, we have 6, 8, 10, and 12. Okay, so here, this is the part of ploid estimation, okay? Now, are you guys there? Okay, I believe so.
Yes.
Great. Let's go to the genotype calling part. This would be easier because we have this grade 2 VCF to SCM. And just let me show you here. And also, if you are running array data, I would strongly recommend to use FitPoly. I think is the best tool for array data. And although SuperMASSA was not developed for sequence data, we have been using SuperMASSA for sequence data, and we have very good results in our hexaploids population. So, and what we are going to do here is to use SuperMASSA with this VCF to SM script, which is a way to use parallelization and also to run several scripts, several SNPs at the same time. And this was implemented by Guy, which is one of our speakers here, with Gabriel and Augusto Garcia from University of São Paulo in Piracicaba. And basically, what you want to do is to just run one command and you have the results, okay? And in this case, just let me run the command first because, and I will explain what we are going to do here. Oops, of course. I need to change the directory. So as always, we need to go to the directory where we have our script. Yeah, there you go. And then just let me run this and I will explain. It's basic. basically copy and paste of this command here, okay? So as I explained to you, we have the Python program, we have the script that we are using with Python. So we are saying here, Python, run this script here with these arguments. And here I use the backslash to make things more readable. I is the input file, minus I, minus O is the output file. And here we have a very interesting thing, a very nice feature, which you can use this plus sign here and the start and end of the files. And in this case, just let me show you this sample VCF. So this is for read VCF files, okay, with dosage calls, with not the doses are not called yet, but we are reading these VCF files to perform the dosage calls, and our output will also be VCF files, okay? And what we can do here is to use these plus signs and say they start and end files. And here we have just samples or small subsets of our hexaploid sweet potato data, right? We have a Burgard in Tanzania biparental population. It's a wonderful population with very, very good data set, and it's publicly available. And this is, if you want the whole data set, just go to my GitHub and everything is there. But I'm just going to run a sample, a small subset. So here we have our 3 files with almost 1,000 SNPs in each file. And we have 1, 2, 3, and we have the start file 1 and end file 3. Here we have the minimum read depth and the maximum read depth allowed. Here, for the genotypes for the offspring, right? Because sometimes you can have multiple individuals and some of those individuals are not belonging to that particular biparental population or panel. In this case, we have the name of the first parent and the name of the second parent. And here we need to point to the SuperMASSA code, because it's using SuperMASSA behind the scenes. Here's the same thing. We have the inference. We can use Heidi-Weinberg. We can use with no structure, we just type ploidy, but in this case, we have a biparental population. Here is the ploid range. And this is interesting because you can say, oh, but sweet potato is an hexaploid. But sometimes there are some SNPs that are outside this hexaploid pattern, and we need to filter out those SNPs. And how we filter out those SNPs? Using this F, and we just using 6 in this case, and just the hexaploid SNPs. Here is just another argument to filter, and so individuals with ploid levels, with those ploid levels that I showed you, lower than 0.8 will be eliminated using this 0.8. Here is to eliminate or to remove individuals with posterior probabilities lower than 7.5. And here we have, if the individuals have more than more than 25% of missing data, right? They will be removed as well. Number of cores or CPUs you're going to use. So if you have a huge cluster with 64 nodes or 64 CPUs, you can use all of these CPUs. And while I was explaining, the results are here. So we have several, since this is raw data, we remove a lot of SNPs, right? And we have some information for the SNPs, but for the information for the analysis, and showing how to read this using MapPoly, which is a program that we will discuss tomorrow. So I just require or a load MapPoly here, and you can use the function read VCF since we have a biparental population in this case. And then we just need to inform the number of both parents. You can also inform the diploid level, but Matplotlib can detect the diploid level as well. And you can just read these files. And here we have the first chromosome. We have here, we have, so diploid level is 6, number of individuals is 350, number of markers, amount of missing data. And here we have the combinations of the dosages in both parents. And you can also plot this to see some information, but we'll discuss this tomorrow. Oh, X1. Okay. Yeah, so there you go. We'll have some information here and we'll discuss this tomorrow. What you can do using some Matplotlib functions is merge all this data. And nope. Let me see here. Yeah, there you go. And then here we have the information for the 3 chromosomes. So you have chromosome number 3, 9, and 12 with about 60 SNPs each one. And if you want to export this to Polymap R, you can use this function here, and then you can go from there in your map analysis as well. Okay. So this was everything I had to show you. So, and if you have any questions, we can discuss if you have time. And thank you for your attention.
Maybe you can comment on the main topics, which were around, the use of SuperMASSA for unstructured populations, diversity populations and things like that.
Yeah, you can also use, there's this publication here. Oh yeah, I forgot to show this. So this is some cytological evidence to show that the diploid level estimation worked in our sugarcane data. But this is the publication where you can see. So in this case, we have, for both scenarios, we have an association panel and we have a biparental population. And the thing is, when you have a biparental population, the genetics behind it is much more solid. It's much more, because you know the founders, you know both parents, and you know the segregation, right? And when you have association panel, you assume you have Heide-Weinberg equilibrium or some of their assumptions, but it's much more like a collection of individuals. So you have less information. And what we observe a biparental population, in this particular case in sugarcane, and that we had this experience, it's much more, the probabilities is much higher in the biparental population instead of the panel. And also, if you have high ploid levels, which is the case of sugarcane, it's very difficult sometimes to distinguish the clusters, because just let me show you this. So if we have like something like this in a dodecaploid, you need it's very easy to have these 2 clusters mixed up. And in a biparental population, it's much easier to see this distinction in these high ploid levels, right? It's doable, right? And it's implemented. This feature is implemented in SuperMASSA, but the results are not as good as the biparental populations one. I think this is the main conclusion.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding in Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.